In this video, we are going to discuss the basic aspects about the categories of randomized clinical control trials, as we have about nine or three categories of the randomized clinical control trial. We start with the parallel trials, in which the randomized clinical control trial is a study in which people are allocated randomly to receive one of two or more of clinical interventions of a drug, of medicine, or whatever it is. Usually, we have two groups: a treatment group, whose participants receive the treatment, which we are interested to find out whether it has therapeutic efficacy, and then comparison or control group for comparing the outcomes. And usually the control can be exposed to conventional practice, the routine treatment which we have, or the placebo, or no intervention at all in some situations. The other groups are called experimental or intervention or treatment group, and the investigator follow up the two groups with a standard manner, and then compare the outcomes in both groups to find out if ever occur or not after the treatment group received the intervention or exposed to the intervention. For example, if a group of investigators uses a parallel design to evaluate the therapeutic effects of a new antiepileptic drug compared with those of a placebo in patients with epilepsy, they would give the new anti-epileptic drug to one group of patients and placebo to a different group of patients. This is a parallel type. Also, we have what we call the multiple arm trials, and this is simple extension of the two-arm trials. Instead of having two arms only, we have more than two arms, maybe three arms, maybe four arms. And here we randomize the participant to more than two groups. For example, in a three-arm trial, the intervention group received two different doses of a drug, while the control group received placebo, for example. And then, despite the benefits of investigating two or more interventions, but here we have to be very cautious, because the statistical power is usually affected and negatively affected. And usually we need, for example, the sample of three-arm trial, we need to increase the trial by 50% to accompany the same statistical power of two-arm trial. And the third type is the crossover trials. In a crossover design, each participant is randomized to a sequence of two or more treatments. Therefore, the participant is used as his or her own control. And crossover trials produce within participant comparisons that we can compare in the same individual, whereas the parallel designs produce comparisons between participants. For example, if we have a participant are randomized to receive either intervention A followed by intervention B, or randomized to intervention B followed by intervention A. And the design is called also two-period crossover design. And here, the crossover trials can be used to investigate, for example, chronic conditions such as cutaneous dysmaniasis, for example, whereas the objective is to investigate the participants' short-term response to therapy. And the condition must also be stable, so that we can ensure at least the basic circumstances when we start our trial are the same for both groups. Clearly, not all interventions in clinical practice can be studied in crossover designs. For example, we cannot study surgical interventions are not suitable for crossover designs, but we can use some drugs, as we have mentioned the example about the treatment used for cutaneous leishmaniasis, for example. The advantage of using the crossover trial is that we will get more precision estimate of the therapeutic effect of the treatment and the trial itself needs a smaller sample size because you are using the in the same sample size at one phase for the as treatment group, and similarly they will be used also as control group. So where the sample size is small compared to the parallel designs, for example. The disadvantage is that usually there is difficulty to avoid any sort of carryover effects that or residual effects of a drug during the first phase of the treatment can be carried over to the second phase of treatment, and this can impair the results. And usually, the crossover trial can be subjected to patient withdrawals or what is known as attrition, and this is will complicate the interpretation and the analysis at the end of the study. And also, there are difficulties that are encountered with the adverse events. There are some drugs, for example, have later effects, therapeutic effects that can be conveyed from the early phase to the late phase and will not appear, will appear also after a period of time. This will impair the results, and usually we encounter difficulties. For example, 100 participants were randomized to follow either a dietary regimen A or dietary regimen 2 for 6 weeks, aiming to find out which is more effective in weight reduction, for example. And the end of this period, participants were crossed over to the other intervention for another 6 weeks, and outcomes were measured every 6 weeks throughout the study. This is a simple example for this. And then the fourth type is the Factorial clinical trials. And here we test the effect of 2 or more treatments simultaneously using various complications, various combinations of the treatments. The simplest factorial design is known as 2x2 factorial design, where participants are randomly allocated to one of 4 combinations of 2 drugs, for example, drug A and drug B, and thus the combination, which we call it FDG, the fixed dose combinations, can be, we give a group of participant A alone, or another group we use B alone, and for another group we use A and B, and then neither A and nor B as control. And this design will allow us to compare the experimental intervention with the control, and to compare the experimental intervention with each other, and to investigate any possible interactions that may occur between them as well. This will enable the investigator to study 2 interventions simultaneously using the same sample size, and the drawback is that, and it is too difficult to ensure that no drug actions have taken place, and this can lead to inadequate interpretation of the results, and as well as biasing of the results. We have also the superiority trials, which is the 5th type of the clinical trials. Here we usually intended to determine if new treatment is different from or better than placebo or existing treatment, active control, for example. And the null hypothesis is that there is no difference between the treatments. And the alternative hypothesis is that the new treatment is different from if you are using 2-sided design, or better than if 1-sided control is used. Now we talk also about the equivalence trials. The aim of the equivalence trials is to prove that there is equivalence between 2 treatments or interventions. Typically, the new drug or device is expected to tally with the efficacy of the current or the standard treatment which are using in our practice currently, for example. And but has the advantages that, for example, despite the 2 treatments are of equivalence, but usually the new treatment may be have some advantages, such as the cost, for example, so it will be affordable for the patient to use, or يعني side effects or quality of lives are better compared to the standard care treatment. And equivalent trials require more patients than a standard randomized clinical control trial. Larger sample is needed. For example, an investigator is planning to test the clinical equivalence of a new antihypertensive drug with the standard antihypertensive drug, which is currently used in practice. The hypothesis is that the new drug is clinically equivalent to if its main reduction, for example, in diastolic blood pressure is 6 to 9 millimeter of mercury compared to the reduction of the current standard treatment, which is 7 millimeter of mercury. The seventh type of the clinical trial is the non-inferiority trials. And here the aim is to prove, to have a concrete evidence that an experimental treatment is not worse than an active control established treatment. And then we determine whether treatment A, for example, of a new drug is at least as good as treatment B, which is frequently established and used in the practice. The hypothesis is that the intervention response, if worse, is still close to the established treatment response, but the detectable difference is too small, but hypothesis is one-sided, so does not require as large sample as equivalence trial, for example. But at the end of the day, the level of evidence is questionable or is lower compared to the equivalence. Here, we have to compare between the three types. For example, we look for the trial type, the purpose, and the outcome. The superiority trial, it shows new treatment is better than control, and the outcome is significant improvement over control. The non-inferiority, it shows new treatment is not worse than control by a small margin. So the outcome is that close to control, but not worse by margin. The equivalence show new treatment is similar to control, and the outcome lies within an upper and lower equivalence margin. Then the S type of the clinical trials categories is the cluster randomized clinical trials. Here, a cluster randomized trial involves social units or clusters of individuals rather than individuals themselves being randomized to different intervention groups. In such circumstances, individual patient level data are also collected. And we have the unit of randomization here, for example, will be the families or the household or work sites or hospitals or towns, entire neighborhoods, communities, etc. And the primary difference between a cluster and patient randomized trial is that participants within one cluster may be more alike or correlated than the participant in another cluster, for example. And the measure of this correlation is known as interclass correlation. As the correlation increases, a trial requires an increasing number of participants. You increase the sample size compared to a patient randomized trial carried in a clinical setting, for example. The last type is Stepped-wedge randomized clinical control trial. Here, all the participants eventually receive the intervention, but the rollout is staggered and the order of implementation is randomized, and it involves sequential transition of clusters such as hospitals or public health units or communities from control to intervention condition in a randomized order and commonly used in public health interventions. These are the main, for example, 9 categories of the clinical trials. And next time, we are going to discuss the other aspects of the clinical trial as a continuation of this. Thank you very much.